Bismillahirrahmanirrahim. We shall explain you today about the chemical composition of the blood-brain barrier. In this regard, we shall explain you the components of the blood-brain barrier. We shall explain you the functions of the blood-brain barrier, and we shall explain you some areas of the brain that lack blood-brain barrier. And then we shall explain you the transport mechanisms across blood-brain barrier, and also we shall explain you the factors that break blood-brain barrier and certain disease conditions which affect the blood-brain barrier and the drug delivery across the blood-brain barrier. So in this context, as you see here in this picture, blood-brain barrier prevents any random entry of the solutes from blood in the brain. Now, as you know, blood-brain barrier, the term barrier is actually used for an obstacle. Here, blood-brain barrier is an obstacle between blood and the brain. So, blood-brain barrier is a physical barrier between the blood and the brain. As you know, our brain is surrounded by blood vessels which supply our brain with the oxygen and the nutrients. This barrier is for selectivity. This barrier prevents the random entry of solutes, large entry of large hydrophilic molecules, and entry of circulating pathogens from the blood into the brain or in the central nervous system. Gases and water passes readily through this barrier, whereas glucose and electrolyte pass more slowly. And the barrier is almost impermeable to plasma proteins and other large organic molecules. Compounds having molecular weight of about 60,000 or having higher molecular weight remain within the circulation, cannot cross blood-brain barrier. Blood-brain barrier permeability is inversely related to the size of the molecules, and blood-brain barrier permeability is directly related to the lipid-soluble substances. In molecular terms, the blood-brain barrier is a continuous lipid bilayer that encircles the endothelial cells and isolates the brain tissues from the blood. This explains how lipophilic molecules, how lipid-soluble compounds can readily diffuse through the blood-brain barrier, whereas hydrophilic molecules are excluded. Blood-brain barrier is formed by endothelial cells of the capillary walls and the astrocytes and feet initiating capillaries and pericytes embedded in the capillary basement membrane. This system allows the passage of some molecules by passive diffusion as well as the selective and the active transport of various nutrients, ions, organic anions, and macromolecules such as glucose and amino acids that are crucial for neural function. The barrier restricts the passage of peripheral immune factors like signaling molecules, antibodies, and the immune cells into central nervous system, thus insulating brain from damage due to peripheral immune events. Now, where from the thought about the blood-brain barrier originated? The thoughts about the blood-brain barrier originated in 1885 from an experiment by which a scientist named Dr. Paul Ehrlich. What did he do? He took a live animal and he injected a trypin blue, a vital dye, a protein albumin binding dye in the vascular system of the live animal. He tried to study that which organs of the animals are colored by this blue dye. What he observed, that the injection of blue dye colored almost everything in the body except the central nervous system. That is, dye colored all the tissues in the body but failed to color central nervous system. So he thought that there must be a sort of barrier between the circulatory system and the central nervous system. This was the initial concept about the blood-brain barrier. As time went on, with better techniques, now we can know a lot about blood-brain barrier. We can talk a lot exactly about blood-brain barrier. Now we can explain what is that structure which prevents the entry of substances from blood to the central nervous system. So cells of the central nervous system cells of the nervous system, including brain, that are responsible for collecting and transmitting messages, is neuron. Cells of the nervous system that are responsible for collecting and transmitting messages is neuron. Neuron is highly specialized tissue. Each neuron is composed of a cell body, a dendrite, and axon. Dendrites that are short antenna-like protrusions that function to receive signals from other cells, and the axons that extend from the cell body and function to transmit signals to other cells. Brain is not only composed of neurons, but it is composed of different type of cells. Approximately 10 raised to power 11 to 10 raised to power 12 are the number of neurons. Communication between the neurons is through electrical signals. But apart from neurons, there are about 10 times more glial cells than neurons. So neurons are approximately 10 raised to power 11 to 10 raised to power 12 are the neurons, and 10 times more are the glial cells. There are 5 types of glial cells. These glial cells includes Schwann cells, oligodendrocytes, microglia, ependymal cells, and the astrocytes. Each glial cell has a specialized function. Glial cells are generally not electrically active, but they are capable of cell division. Glial cells occupy the spaces between the neurons and provide them some electrical insulation between them. But here, our focus is on transport of metabolites in and the out of the cells. In this context, astrocytes play an important role in this function. The astrocytes send out processes at the external surface of the central nervous system. These processes are linked to form anatomical complexes that provide sealed barrier and isolate the central nervous system from the external environment. Astrocytes also send out similar process to the circulatory system to seal it off in a selective manner. Astrocytes induce the endothelial cells of the capillaries to become sealed by forming tight junctions that prevent the passive entry into the brain of water-soluble molecules. These tight junctions form what is commonly called as blood-brain barrier. So astrocytes induce the endothelial cells of the capillaries to become sealed and form tight junctions. And these tight junctions, they even prevent the passive entry of into the brain of the water-soluble molecules. These tight junctions are commonly called as blood-brain barrier. Water-soluble compounds enter only if there is a specific membrane transport system for them. So neuron is highly specialized tissue. In the central nervous system, neurons are master regulatory body. Neuron is very sensitive. Neuron likes their private environment. They like a very stable environment. Therefore, everything from the blood is not reaching in the neuron because of this barrier. Now question is, why is this barrier? Although neuron gets nutrition from the blood and neurons give their waste products into blood, but still all changes are still all fluctuations of the blood chemistry does not reach to neuron. For example, hormonal changes occur in the blood very frequently. For example, during fasting period, hormonal condition is different in the blood, and similarly, hormonal composition of blood is different in the postprandial condition. Hormonal condition in the blood is different during physical activity, or it is different in the conditions of stress. So biochemical changes in the blood occur very quickly, but neuron is protected from all these changes in the blood. Neuron does not like that every change which occurs in the blood should reach to neuron, and neuronal changes should not be at the mercy of changes in the blood. So one of the function of blood-brain barrier is to provide a stable environment to neuron and protect the central nervous system from fluctuating hormonal levels in the blood, fluctuating metabolite level in the blood, and fluctuating ionic levels in the blood. Then coming to the components are the structure of blood-brain barrier. Now, blood-brain barrier, it is composed of blood-brain barrier endothelial cells of the blood vessels. Blood-brain barrier, what is the composition of the blood-brain barrier? It is composed of blood-brain barrier endothelial cells of the blood vessels. These blood-brain barrier endothelial cells of the blood vessels are surrounded by pericytes. They are surrounded by glial cells, pericytes. They are surrounded by astrocytes through their foot processes. The basement membrane of the blood-brain barrier is composed of extracellular matrix components, including type 4 collagen, protein lamellin, heparin sulfate, and the protoglycans. All these constitute a continuous layer that separates the blood vasculature from the neuronal tissues. All these components constitute dynamic functional unit, and that functional unit is called neurovascular unit. So blood-brain barrier, it is composed of blood-brain barrier endothelial cells of the blood vessels, which are surrounded by pericytes and the astrocytes, and basement membrane is composed of extracellular matrix compounds, and all these components constitute a functional unit, and that functional unit is called neurovascular unit. Blood-brain barrier endothelial cells of the blood vessels, they constitute innermost luminal component of the blood-brain barrier. And blood-brain barrier endothelial cells have many unique properties that separate it from other peripheral endothelial cells. For example, the blood-brain barrier endothelial cells of the blood vessels, they contain higher number of mitochondria, allowing generation of greater amount of biological energy, which is required for the maintenance of the blood-brain barrier integrity, and they increase its selective molecular permeability. So blood-brain barrier endothelial cells of the blood vessels, they have higher number of mitochondria, which is not seen in the other endothelial uh, cells. And blood-brain barrier endothelial cells are polarized and display numerous receptors, ion channels, surface transport proteins, along with limited vesicular transport. They also possess extremely low level of leukocyte ADN molecules, which stop or which hinder the infiltration of immune cells into central nervous system, and they possess a negative charge, and this negative charge on the surface repels the negatively charged compounds. All these features regulate solute permeability into the central nervous system along the junctional complex. Then here in this picture, we compare the endothelial cells of the capillaries in the brain, which are entirely different from capillaries of the other tissues, because these endothelial cells of the capillaries in the central nervous system, they provide major function of the blood-brain barrier. So endothelial cells of the capillaries in the brain are different from the endothelial cells of the capillaries in other tissues. Now, capillaries are in general, we are talking in general, capillaries are of three types. Some capillaries, they are called as continuous capillaries. Some are called as fenestrated capillaries, and some are called sinusoidal capillaries. So what are continuous capillaries? Continuous capillaries are those where endothelial cells of the capillaries do not have any junction between them. It has basement membrane around it that is also continuous, and there is no gap in between them. Such capillaries are called continuous capillaries. Then the other second type is the fenestrated capillaries. Fenestrated capillaries are those which have pores, or which have windows, or which have holes. Through these holes, certain things can pass very easily, and these holes are called as endothelial windows, or they are called endothelial fenestrations. And third type is the capillaries, is the sinusoidal capillaries, which are also called as broken capillaries because there are larger gaps, and such capillaries are seen in the liver or in the spleen. In this regard, the endothelial cells surrounding the capillaries in the central nervous system, they have interendothelial gaps, but those interendothelial gaps are stitched tightly. They are stitched tightly. So there is no gap. And how they are stitched? Their stitching thread is actually the proteins. And these proteins are named as claudin and occludin. What is claudin? Claudins, they are primarily sealing proteins of tight junctions. And occludins are acting as signaling processes, and there are junctional adhesion molecules. Tight junctions reduce the permeation of polar solutes in the brain's extracellular fluid and limit the passage of proteins. Tight junctions are composed of series of transmembrane adhesion proteins, cytoplasmic proteins, and actin. Most important thing present in the central nervous system, which acts as blood-brain barrier, is tight junctions in the endothelial cells lining the central nervous system. So actually, these tight junctions, they are actually the blood-brain barrier. And then basal membrane is composed of extracellular matrix components like type-4 collagens, laminin, heparin sulfate, and protoglycans. And disruption of basal membrane will lead to damage to tight junctions and blood-brain barrier integrity. So, what is blood-brain barrier? At the interface between the blood and the brain, endothelial cells are stitched together by tight junctions, which are composed of smaller subunits, frequently biochemical dimers that are transmembrane proteins. We explained you they are claudin, occludin, and junction adhesion molecules. Claudins are primarily sealing proteins of tight junctions, and occludin acts as a regulator for signaling processes. Each of these transmembrane protein is anchored into endothelial cells by another protein complex that includes zonula occludins and associated proteins. Blood-brain barrier is composed of high-density cell restricting passage of substances from bloodstream much more than the endothelial cells in the capillaries elsewhere in the body. So blood-brain endothelial cells of capillaries, they have unique properties. We have already explained you. We told you that blood-brain barrier endothelial cells of the capillaries, they have large number of mitochondria. Mitochondria are powerhouse for generating the energy required to maintain the blood-brain barrier integrity and augment its selective permeability. So they have blood-brain barrier endothelial cells of the capillaries. They have one separate character that they have higher number of mitochondria, and they are polarized, and they display numerous receptors, ion channels, surface transport proteins, along with limited vesicular transport. These characters are not in the endothelial cells of ordinary capillaries, and they have extremely low level of leukocyte adhesion molecules, which hinder the infiltration of immune cells into the central nervous system. These are the important characters of the blood-brain barrier endothelial cells of the capillaries. Then basal membrane, we told you, is composed of the extracellular matrix components including type-4 collagen, protein laminin, heparin sulfate, and the protoglycans. Disruption of basal membrane will lead to damage to tight junctions and the blood-brain barrier integrity. Then there is important role of the glial cells. That is, these includes pericytes, astrocytes, and microglia. In the beginning, we told you there are approximately 10 raised to power 11 to 10 raised to power 12 are the neurons, and 10 times more are the glial cells. So pericytes, they are embedded within the basement membrane, and they prevent leukocyte infiltration into central nervous system. Astrocytes are the major glial cells of the neurovascular unit. Astrocytes completely cover the cerebral blood vessels with their end foot and provide sealed barrier. Interaction between astrocytes and neurons determine synaptic transmission. They also serve as stem cells during development. And microglia are most abundant immune cells of the central nervous system. They are primarily mediators of central nervous system inflammatory response. Microglia cells detect the very first sign of the tissue damage. Then transport across the blood-brain barrier. Despite the efficient barrier function for preventing the entrance of non-desirable compounds, healthy blood-brain barrier also act as filter and allowing selective exchange of solutes and regulatory factors between the blood and the brain. The transport across blood-brain barrier can be divided into two main pathways. That is, one is paravascular pathway and other is the transcellular pathway. So we have mainly two pathways for transport across the blood-brain barrier. We told you that this blood-brain barrier is efficient. It is functioning as filter, or it is for allowing selective exchange of solutes or regulatory factors between the blood and the brain. But apart from this, again, it has two transport systems. One is the paravascular and other is the transcellular. In the paravascular transport system, it occurs in the brain here by passive diffusion of low molecular weight hydrophilic molecules through tight junctions into endothelial cells lining the central nervous system. But this is negligible. So, in the brain, paravascular transport, it occurs in the brain by passive diffusion. So, by process of passive diffusion, low molecular weight hydrophilic molecules can pass through the tight junctions, but definitely it is a very negligible. Then there is transcellular transport system via blood-brain barrier, endothelial cells of the vessels, and this is two types. Transcellular transport system, it is either energy dependent active or it is not energy dependent, it is passive. Passive transcellular pathway includes transcellular diffusion that takes place at the luminal membrane of the blood-brain barrier endothelial cells, and it is only restricted to gases such as transport of oxygen and carbon dioxide. It takes place through passive transcellular pathway. Active transcellular pathway of transport, it is again of three types. One is receptor-mediated transcytosis, other is active efflux transport, and third is the carrier-mediated transport. Receptor-mediated transport, it is the main pathway used for the uptake of molecules that do not possess a specific carrier, such as by this receptor-mediated transport, there is transportation of hormones and high molecular mass proteins. These molecules bind to specific receptors on the cell surface, resulting in the formation of endocytic vesicle that will cross blood-brain barrier to release ligand and allowing the receptors to be recycled. Example of receptor-mediated transport is transferrin receptors and the LDL receptors are the examples of receptor-mediated transport. Then the second mechanism is active efflux transport. Efflux pumps are responsible for removal of substances out of the central nervous system into systemic circulation. Among the efflux pumps, we found proteins that belong to ATP-dependent binding cassette transport superfamily. And then third transport is carrier-mediated transport. Carrier-mediated transport can be either active are passive, and it allows the exchange of molecules between blood and central nervous system through blood-brain barrier via specific via specific transporters like glucose transporter 1. There is large neutral amino acid transporters and nucleoside transporters are among the carrier-mediated transport proteins, and they are of great importance in the blood-brain barrier homeostasis. Here in this slide, we shall explain you the differences between the blood-brain barrier versus capillaries of peripheral nerves. In this context, as you see here, tight junction resistance in blood-brain barrier is about 5 to 10 ohm per square centimeter, while tight junction resistance in capillaries of peripheral nerves is about 2000 ohm per square centimeter. Then there are no fenestrations in the blood-brain barrier, but fenestrations are present in capillaries of peripheral nerves. Vesicular transport is deficient in blood-brain barrier, but vesicular transport is abundant in the capillaries of peripheral nerves. Pinocytic vesicles are rare in blood-brain barrier, but pinocytic vesicles are abundant in the capillaries of peripheral nerves. Mitochondria are abundant in the blood-brain barrier, but mitochondria are not abundant in capillaries of peripheral nerves. Then, tight junctions, selective transport, and astrocyte foot processes are very high in blood-brain barrier, but are rare or absent in capillaries of peripheral nerves. Then coming to the functions of the blood-brain barrier. Blood-brain barrier restricts the passage of pathogens, harmful metabolites, and large molecules are the hydrophilic molecules. And blood-brain barrier protects the central nervous system from the fluctuating hormonal levels, metabolite levels, and fluctuating ionic levels in the blood, because neurons love to have very stable level of electrolytes and protons. So one purpose of the blood-brain barrier is that it acts as the stabilizer. It stabilizes the internal environment of the neuron or internal environment of the central nervous system. Then another function of the blood-brain barrier, the blood-brain barrier is good holder of the neurotransmitters within the central nervous system. So blood-brain barrier is good holder of neurotransmitters within the central nervous system, so that neurotransmitters should not flush out in blood. So as you know, how much blood passes through the central nervous system? About 800 ml per minute. It is about 800 ml per minute, but these neurotransmitters do not flush out in the blood. So one of the important function of the blood-brain barrier is that blood-brain barrier prevents the free movement of certain substances between the peripheral blood and the central nervous system environment. Then blood-brain barrier allows the diffusion of oxygen, carbon dioxide, hormones, and small nonpolar molecules. This barrier, blood-brain barrier, also restricts the passage of peripheral immune factors like signaling molecules, antibodies, and immune cells. blood-brain barrier is insulating the brain from damage due to peripheral immune events. All these are the functions of the blood-brain barrier. Then, not all the areas of the brain has blood-brain barrier. What is circumventricular organ? There are some areas in the brain where blood-brain barrier is weak and allows the substances to cross into brain freely. This is known as circumventricular organ. So, the areas where blood-brain barrier is weak, which allows the substances to cross into brain, these areas are known as circumventricular organ. These individual structures are located adjacent to fourth ventricle in the brain. So, circumventricular organ, they are located where? They are located adjacent to fourth ventricle in the brain and are characterized by dense capillary beds with permeable endothelial cells, unlike those cells of the blood-brain barrier. And this circumventricular organ, these sites are actually for the bidirectional blood-brain communication for neuroendocrine function. Included among the circumventricular organs having highly permeable capillaries are the area postrema, subfornical organ, median immense, pineal gland, posterior lobe of the pituitary. Pineal body secretes melatonin, posterior pituitary release neurohormones like oxytocin, vasopressin. Median immense connects hypothalamus with pituitary. Subfornical organ are chemoreceptor areas important in regulation of body fluids. Area of postrema is vomiting centers. So this is circumventricular organ where blood-brain barrier is weak and allows substances to cross into brain freely. Then coming to the not all vessels in the human brain exhibits blood-brain barrier. There is also blood cerebrospinal barrier. A barrier similar to blood-brain barrier may exist in the choroid plexus, and this barrier in the choroidal plexus is called blood cerebrospinal fluid barrier. Blood CSF barrier is the function of the epithelial cells of the choroid plexus. Blood CSF barrier is function of epithelial cells of the choroidal plexus. The idea in between these two barriers is to inhibit both blood-brain barrier and the cerebrospinal fluid barrier. These two barriers, they inhibit entry of harmful substances in the brain, but the difference between two is that blood-brain barrier is an endothelial barrier, whereas blood CSF barrier is epithelial cells barrier. Your question is where tight junctions are located. In case of blood-brain barrier, tight junctions are located in the endothelial cells, whereas in the cerebrospinal fluid barrier, tight junctions are located in the epithelial cells. So they both prevent the unwanted substances in the brain. Then coming to breach of blood-brain barrier. Blood-brain barrier can be broken down by high blood pressure. High blood pressure opens the blood-brain barrier. Then blood-brain barrier is not fully formed at the birth. Blood-brain barrier appears to be functional by the time of birth, but blood-brain barrier is not fully mature at the time of birth. For example, we take the example of the unconjugated bilirubin. In the newborn babies, if unconjugated bilirubin level increases about 20 milligram per deciliter, it can cross blood-brain barrier and causes toxic encephalopathy. But in adults, if unconjugated bilirubin level goes above 20 milligram per deciliter, it cannot cross blood-brain barrier. From this example, we conclude that blood-brain barrier is present at the time of birth, but it is not fully formed at the time of birth. Then the other factors which can breach the blood-brain barrier, that is the high concentration of substances in the blood, can open the blood-brain barrier. That is hyperosmolarity can cause damage to blood-brain barrier. Then exposure to microwaves can open the blood-brain barriers. Then exposure to radiations can cause damage to blood-brain barrier. Exposure to infectious agent can damage to blood-brain barrier. Trauma, ischemia, inflammation, pressure injury, they all can cause damage to blood-brain barrier. Then coming to diseases affecting the blood-brain barrier, neurological diseases such as encephalitis, multiple sclerosis, brain traumas, Alzheimer's disease, epilepsy, stroke, tumors, they all can cause drug delivery and the blood-brain barrier. The blood-brain barrier is permeable to small and lipophilic fat-loving molecules up to 800 atomic mass units. But larger molecules are not transported across unless there is an active transport system available. So only lipid-soluble substances can pass from blood into the brain. Higher is the lipid solubility of drug, more will be the entry into the brain, and more will be the effect of drug on the brain. Lesser is lipid solubility of drug, lesser will be its entry in the brain. If the drug is not lipid soluble, it cannot enter the brain, will have no effect on the brain. For example, aminoglycosides like streptomycin, gentamicin, amikacin are not used in the treatment of patients suffering from meningitis. If you are using it by intravenous route, it will not move into the brain. It will not penetrate in the meninges. Why? Because aminoglycosides are highly ionizing molecules, highly polar molecules. More is the ionization, lesser is the entry into the brain. That is why, in case of meningitis, which is due to gram-negative microorganisms, still we cannot use aminoglycosides. If you want to use aminoglycosides, you should give it direct in the spinal cord. In Parkinson's disease, there is deficiency of neurotransmitter dopamine in the brain. But you cannot give dopamine to the patient suffering from the Parkinson's disease. Why? If you are using dopamine by intravenous route, it will not pass to the brain because dopamine is typically a water-soluble product. That is why we use precursor of dopamine, that is levodopa, because it is fat-soluble after entering in the brain. The levodopa undergoes process of decarboxylation, and levodopa gets converted into dopamine. And this is the real effective drug in the patient with Parkinson's disease. So this is all about the drug delivery. With this, we stop our lecture about blood-brain barrier.